Today, we are going to talk about exploratory data analysis, and we're going to talk a little bit more about summary statistics and what to do with the material that we have generated. Okay, Beepsa, if you could take a look, it seems like genomics still is being shown as paid. Okay, thank you. Yeah, so hopefully you're getting a sense of how these different technologies are going to work together. And I will hopefully demonstrate some elements of what we have been doing on the platform, why platform and external server is a beneficial solution as opposed to trying to do it on your computer, just simply not going to work very well. Even if we just take some of these big data sets that we are analyzing right now. So hopefully we can discuss that. But before we go into the topic for today, let's take a look at your progress. I've been checking, and Beepsa has been also checking up on everybody's progress. And I think it is going really well. So a lot of you have started the pipeline. You can see right now, some of you are starting to have the points for the pipelines. I do want to again just mention a couple of you still have not fixed your names. And I think that as you complete the courses, just think about the the certificates that you're getting. So that whole name is what is going to appear on your certificate. So please just make sure that you update your name to be the real full name that you would like to be there. Also, I saw that at least one person has submitted on the forum the reply to the assignment. Thank you, Marek. And hopefully we'll discuss this assignment a little bit more. I know that it was a little bit rushed last time when we had to go into the code, and it might have not made a lot of sense how to do it. You might have gotten confused, so hopefully we'll address that today. Let's talk about what we discussed last time, which was the RNA-seq pipeline. So let's just see how everyone remembers what we talked about and what is it that we were trying to measure. So measuring mRNA abundance in a sample could be done with a number of different technologies. For example, when we talk about reverse transcription PCR or RT-PCR, what it does is it amplifies DNA based on two primer sequences framing into section of interest. And since PCR amplifies DNA, not RNA, RNA is first converted back to DNA prior to amplification. This is done via a process called reverse transcription. So that's where RT-PCR comes from. In nature, this is a process that is carried out by retroviruses. Then we talked about northern blotting, which is when an RNA sample is digested into fragments that are separated by size in gel using electrophoresis. And they are then transferred to a membrane that can be probed to test for the presence of a hypothesized RNA transcript. So again, you have to specify which RNA transcript you are interested in. And then we talked about briefly microarrays, which are like northern blots, are hybridization based, which means that they have primers in these slots. And unlike northern blot, a microarray yields highly detailed information about the individual genes that have been transcribed. So these signals are then detected, quantified, and used to create a digital image of the array. And then the digital image is used to identify the transcribed genes and how many of those are transcribed by color and by the amplitude of the color. So some of these technologies have been around for a while and they're still used today. And so a lot of data from these different technologies is useful and oftentimes used regularly in labs to test for mRNA abundance. So whether it's present and how much of it is present. And then the next generation sequencing approach was developed more recently and has since been tremendously improved for accuracy, length of reads, quality and quantity that these machines can produce. And as a result, also the prices went down. And so one of the popular techniques to quantify mRNA without any kind of targeted sequences that we're looking for, so just kind of looking at everything that is being expressed, is next generation sequencing based on the Illumina protocol. And as you remember, what we discussed is one of the challenges is really having this detailed process of extracting the useful information from all of the sequencing reads. Now, the information for next generation sequencing is stored in these three types of files. So typically in a pipeline called RNA-seq, or if you're doing whole genome sequencing, you can also you will also use the same kinds of files. Basically, the read information is stored in the FASTQ files. The reference genome is stored in the FASTA and GTF files. And we also talked about the actual process of sequencing, how as a result of amplification and preparing the data for analysis, you should expect some pre-processing steps to make sure that the information that you are extracting is actually based on just the biological information, not the technical information that was produced as a result of the sequencing process. So yesterday we started this pipeline. I saw that one person's pipeline actually finished, and that was as a result of a mistake. It should not have finished. So most of you should see something like 50 to 66% done. Have you checked on your pipelines? Can you tell me what is the percentage done for your pipeline? The way to check it, by the way, is if you go back to your profile under, okay. So you will go, yeah, under my pipelines, and there you should see, okay, great. So a lot of you have already 66. Now you could have, used a number of different combinations. And please, for the rest of you, also let me know how much you have left. But basically what the pipeline calculation is how many jobs in total, and then you divide the number that has completed by the total, and that's how you get the percentage. So did anyone else get their pipeline started and has the percentage? Okay. So Anita, are you sure you started your pipeline? Is it green? If you go there, is this bar green? It just says 0? Yeah, if you just stay on the page, it's not going to move. You have to refresh the page to see the progress. Okay, Emeka's finished. So Emeka, make sure that you actually have an output. Okay, another thing that I want to point out here is that you can see what data has been uploaded. So remember, we use this SVL file. So if you can go into your pipeline, just click right here on uploaded data. If your pipeline is more than 0%, then you should see here the files that were uploaded. Okay, so please let me know if you can see files here on uploaded files. So if you can, say yes. Does anyone see files in there? Anyone else? Emeka, do you see uploaded files here? And also, Marek, if you could tell me, I know that I think it's your pipeline that says that it has completed, but please check if you see any uploaded files here. I think your pipeline had a problem. Okay, great. But Emeka, do you see uploaded files? All right. All right, excellent. So just making sure, you can also see here how much space is being used just for this pipeline. Okay, so you can see that the pipeline has not finished, but it already is using 122 gigabytes of space. And the reason for that, right, if you look over here and uploaded data, you can see here how many bytes is each one of these. So it's not just the files themselves. What the pipeline does is it takes all the files and loads them here, saves them, converts them into cleaned up reads, saves them, converts them into SAM and BAM files, which are also large because they contain all of the information about the reads, saves them, et cetera, right? So this is a multiple of whatever files you uploaded. And the reason that typically this type of data is analyzed on some kind of a server or on the cloud is because of these huge file sizes. Now you can, of course, remove these files, but remember that we only have really 10 files in here, right? So this is not even a large experiment. If you look online, on NCBI, for example, some of these experiments have hundreds of samples. So you can imagine how larger pipelines are going to become after you analyze real data sets from these large, for example, human RNA-seq. You know, the genome is large. You have over 20,000 genes. Whole genome sequencing might be even larger. Okay, so that was about the pipeline. And the overview of RNA-seq, as you remember, the logical steps that we took is we took pre-processing steps, which were PCR duplicates and trimmomatic. We did mapping, and there are different mapping strategies that you could use here, either a more detailed one on all of the genome or just on the transcripts, which are called the transcriptome, and then quantification of expression levels. Again, you have a few different options. Now, some more recently developed methods seek to reduce the memory usage and do what's called alignment-free quantification. Those include salmon and sailfish. And so we have those also on the platform, and you can try and work with those. Those will only give you quantification of transcripts that are known. So they do not look for actual genomic variation. They look only at known transcripts. Expression table, one of the things that we're going to be looking at today is a table of genes, which you see in your rows, and by sample, we know which gene has what kind of an expression. So if you did the assignment for yesterday, you probably noticed that visualizing the gene expression gives you a bar for each one of your samples. And the level of expression is that number right there. And if you look at a profile of a gene, what we call a profile of a gene, you can tell what gene it is by the name. For example, we used Ensemble ID. If you are working through Transcriptomics 1, there's a data set there that contains genes from both mouse and human. In our case, for cell lines, we're only using human. But here you can see that the extension ENSG or ENSMUS-G gives you a sense of which reference genome was used to quantify that gene expression. Okay, so to see that you all understood and have a good familiarity with RNA-seq, let me ask you a few questions. So what type of files are used as input for the RNA-seq pipeline? One, FASTA or FASTQ files, which include DNA sequence data. Two, SVL or URL files, which include RNA sequence data. Three, RSEM or TXT files, which include our DNA sequence data. Four, Excel files, which include RNA sequence data. Okay, so Deepika, I'll get to your question here in a second. Okay, take a look at the full statement. Don't just focus on one part of the statement. This is supposed to be a little bit tricky. So don't just think, okay, what did I do? Think also what was in the data. Okay, so the correct answer is number one. Why? Well, because when we are sequencing the data, we are converting the data to cDNA. And so you can see here it says FASTA or FASTQ files, which include DNA sequence data. And that's the problem with number two. We did use an SVL, or we can use URL files, but it will always include DNA sequence data. Never will you have actual RNA sequence data because it is converted to cDNA. And obviously these other ones, I don't believe anybody mentioned those. So great, you guys understand. Now, another thing to remember is that when we are sequencing, let's say we're looking for RNA, then we are trying to interpret what is in our data as a result. And so there are going to be several types of interpretation approaches. One is you can obviously just convert it to a common gene name from Ensemble ID. Two, you can look for that common gene name in terms of how it is included in different gene ontologies, pathways, or even location in the cell. And so you can see here the location in the cell, cytosol, nucleus, where these different processes happen. You can further define them after you look at the gene property. Okay, so let's address these questions. So Deepika asked if there's a pipeline available that can run a quality control test for a SNP dataset, then we can just provide our raw data, and as output, we get processed data. Okay, so let me clarify. For SNPs, so SNPs are single nucleotide polymorphism. So this would come from a DNA sequence data. So if you're looking for variation at the DNA level, you might want to characterize mutations or SNPs. So SNPs are what we call single nucleotide variants, but that typically occur in many samples in that particular location. So if we're looking to find single nucleotide variants per sample compared to the reference genome, the question here, can we run quality control tests? So there's not really a quality control test, it's just the detection of that SNP. So if a SNP is a SNP, is reported as a SNP, typically that is based on many different samples. And the reporting is that it occurs in a certain percentage of those samples. Now, what is the raw data? The raw data are individual reads. And you can do, and you do in any of these pipelines, you do first quality control of the reads. In fact, Bowtie internally has a quality control script involved in first looking at the quality of those reads, and all of the quality that have low, or all of the reads that have low quality are discarded before you actually do the mapping. Now you get statistics on how many reads were mapped and the quality of mapping in one of the outputs. We will look at that tomorrow when these pipelines have finished. Okay, Deepika, did I answer your question? Okay. Smitha, how to convert FASTA or FASTQ files to SVL. So SVL, again, let's take a look again at SVL. What is inside that file? So here we have samples.svl. And it's really just a link. So it's just a link to the FASTQ file. So there's not any conversion. This is just, I uploaded these files using FTP, put them in a folder, and now I have a folder, right? This is the folders, and I can have this. Just if you do it on your computer and you want to reference it in R, you would also use some kind of folder structure. Okay, why should we do this conversion? Well, how would I give you a bunch of these files? These files are big, so I'm not going to email them to you. Instead, I upload them on the server where all of you can use it from that single location. Does that answer your question? All right. Okay, so let's go to another question. Which of the following are pre-processing algorithms? 1, top hat and D-seq, 2, PCR clean and trim-o-matic, 3, bow type 2 or BWA, 4, edge R or D-seq 2. All right, excellent. So what do these actually do? You guys are all right. Trim-o-matic cleans technical sequences, PCR clean removes PCR duplicate. So I see that all of you got that part correct. So now let's talk about the output from this pipeline. So hopefully soon it will finish. Once we get that, we will take a look at how the data looks. How do we characterize or describe this data? And then how do we prepare that data for downstream analysis? And how do we deal with some of the issues? So here are some of the things that we want to cover today: summary statistics, visualization, missing data and data errors, and summary statistics in R. So we'll do some practice in R as well. Okay, so what are we going to be dealing with? This is a data set that is so-called raw data, right? So this is direct output from that pipeline in FPKM or TPM. So again, we have sample name and we have gene ID, and here we have expression level. Now, let's take a look at this file. This file is, again, in the same link that you got yesterday from BIPSA. So you can go back and open that folder, and inside there are a number of files. So let's take a look at some differences between these files. So this file we used, and it's really just simplified, right? In terms of ID, it has names of groups. It doesn't tell you the actual name of that sample. And instead of actual genes, it just tells you gene 1, 2, 3, etc., right? So one of the things that we asked you to do was to visualize gene 10, or to compare different genes and see which one is more informative or less informative. So we're going to talk about this just in a second. But another file that is in here is what's called the raw output. So here you can see cell lines, expression, FPKM. So let's open up this file, and I'll just open it in Excel for now. So let me ask you, tell me yes if you have found this file, cell lines, expression, FPKM. Okay, great. So again, cell lines, expression, FPKM. Okay, just 2 people. Has anyone else found the file? Okay, Mark, Krishna. Anyone else? Just give it a couple more people. Sumitha, great. All right, great. So again, doesn't matter where you open this in. You can open it in Google Docs, you can open it in R, you can open it in Python, whatever you like to work with. The point is we want to take a look at what is inside. So inside, we have these are gene IDs, right? And here we have the name of our sample. The actual name of the sample is this right here. This is for sequence read archive ID. And then this was added by the pipeline because adapters were removed and PCR cleaning was done. And this was a pair end file, right? So all of your information is stored right here. Now let's go and see how many genes do we actually have. So how many genes do we have in total? How many genes do we have in total? Okay, 19,990, right? So about 20,000 genes. This is a typical number, right? So 20,000 gene is a typical number that you would get, but obviously not all of these are going to be useful information. How do we know that it's not going to be useful information. We can create here a column, and let's call it max, and we'll just calculate the maximum number from all of our samples. Okay, so here, for example, the maximum number is 20. Let me make it for all of my samples or for all of my genes. And then I can here make a filter, right? And let us just look at what are some of the numbers that we have. So you can see that the numbers start from 1, and which means that this is already a filter, right? So there's not a single gene here. Why don't we have 20 or more? Because there were probably just a few that were 0 expression, right? And that happens also if you take the ultimate raw output, you will typically have a number of genes that have 0. Now, is 1, let's take a look at one of the genes, or some of the genes, that have the maximum of 1, right? So how do we make a bar plot? Here, let's make a quick bar plot and take a look. Okay, so here I have a bar plot. You can see that, well, I guess that's fine. That's not how, here, let me get it in a different format here. Okay. Okay, great. So here I have my regular bar plot, and I'm just going to quickly do this. Okay. So here I have my profile. Now, let's assume I am using this information from these gene expression profiles to characterize my samples. Typically, a characterization of the sample includes a comparison to something else. Let's say I have a control here and some condition. Doesn't matter which ones are which. If I take any average between these, will I ever find any kind of a significant difference between groups of genes, right? And the question is, what is significant, right? So in this case, because my range here is from 0 to 1, whatever the difference I will get, it is always going to be less than 1, right? And since I have almost all of my samples represented in between this variance of 0 to 1, I will never find any significance, because an expression level overall should be at least a number that gives me some kind of a meaningful difference, right? So in this case, this is never going to be meaningful. Even if I take 2, it's never going to be meaningful. And typically, that's why before moving down with other types of analysis that you do, you might want to filter this out by a certain number. Okay, but before we jump into that, let me clear this filter now. So before we actually jump into that, there's another problem. The other problem is that if I take all of this information and I draw a histogram, what is this histogram telling me? This histogram is telling me about distribution of all of my values for a particular sample. And it doesn't matter which sample I take, okay? The data will look like this. Who can tell me what is the problem with this histogram? Or not what is the problem, but how would we describe or characterize this distribution? Okay, it's skewed, right? It's skewed. What is skewness? Skewness means that the data is not normal, right? So a normal histogram, if I take what I expect to be a normal histogram and what is typically going to be used in many kinds of downstream analyses, whether it's comparative analysis or dimensionality reduction analysis, I will always expect, or these algorithms will expect, a normal distribution, which is going to be like a bell curve, which means that most of your values are in the middle. So the majority of your values are going to be symmetrical, and the middle represents the most typically observed values, right? So why is that symmetry useful? That symmetry is useful because I can now work with properties of this data, like mean or standard deviation, or take some properties based on this normal distribution. In this case, I am not going to be able to rely on a lot of these properties. So if I take the mean is going to be very low, and you can see here the bins that I have are between 0 and 39. That includes the majority. Point 1 includes 17,000, right? So this means that point 2, which is between 117 and 156, right? But that means that some of my data over here will still have some information, right? And we can't really see here, but some of the information that I have is going to be way over there. And so the way that we can estimate how informative this is going to be for different types of statistical analysis, and a statistic is going to be derived from these characteristics of your data, is if we plot a bar plot. So instead of a histogram, we can quickly here do a bar plot. So let's take a look at what the bar plot will summarize those histograms across all of our samples. And the bar plot explains a few things. It explains the distribution of your interquartile range, or 75% of your data, and shows you outliers, including the mean and things like that, but it also shows you outliers. And we can see that the majority of the data are outliers, right? So the majority of the data over here, right, the 75% of the data are in the boxes, which we can't even see, and then some of the information are the outliers, and that is the most prominent. So whenever we start doing comparisons, we will see that work against us, right? Because the most informative, the most prominent, strong signal in our data actually represents a very small minority of the data as a whole. So what can we do about that? What we can do about it is we can look for a way to normalize this data and make it more like normal distribution. Okay, so what we are looking here is distribution of data, how distributed our values are. And to do this transformation, typically for many types of biological data, people transform it to a different scale. Okay, and so this is called log normal transformation. What is log normal transformation? Log normal transformation is just looking at the histogram of your raw data, okay, and making your actual data more visible, right? So right here, this is a box plot, and in a box plot, you see these statistical properties for each one of your sample. This is the interquartile range, and what you want, for example, in differential gene expression or in a comparative analysis is to compare your averages between groups of samples, right? So those averages have to be a reliable statistic for you to compare. So here we have our raw data that looks like this, and the majority of values are not seen very well, but the outliers would be seen. And so if we do this and we actually do log transformation, this will stretch out the distribution, because the scale of the very small values, which are the majority, will be much closer to the scale of the large values. And so we have stretched out the information that was here in these boxes, and now we see our median much more clearly. Okay, so how do we do this? We actually do logarithm of the value +1. Okay, so what happens in the case of non-parametric analysis. That's true. We can do all kinds of analysis on raw data, but the problem is that in many cases, as you explore this data, you will rely on this normal distribution. And you can do tests on non-normalized data, but in many cases, right, before you actually do your test, you have to explore the data. And to explore the data, you are trying to look for significant signal, right? So before tests, before you actually do a test, you have to explore your data. And to explore your data, you actually have to understand what types of patterns are present in your data. And when your data is non-normal, to find those data, to differentiate between significant and insignificant patterns is much more difficult, right? And we'll see the impact of that when we try some of these analyses in future sessions. Maybe in our session tomorrow, we'll actually start seeing that. So when we take a look at log scale transformation, let's just do a quick example first in Excel, and then I'll show you how to do this in R. We already saw here the histogram of this sample, right? So let's do the same over here with log scale transformation. So let's just take this sample, right? And we'll just do here ln, which is normal logarithm of this number, and we're going to add a 1. Why are we going to add a 1? Because if we have a 0, that is infinite. Logarithm of 0 is infinite, so we can't do that. And so we are going to add 1. And logarithm of 1 is 1, so it's not going to change anything, right? So what will happen is that, and we can do this for all of our 19,990 genes, if it will let us do that. Maybe not. Okay, so here's an example, by the way, right? So this is a 0, and this is a 0, right? So logarithm of, okay, so let's see. Mm-hmm. There we go. All right, so now we have our data. Let's try and look at what happened with our distribution. Okay, so here we are starting to see the improvement of this, right? It is still not normal, right? We don't see a bell curve, but the information is starting to become more visible. Okay, so what is in your typical histogram? In your typical histogram, what you see is the amount of information that each part of your data contains, right? So here, for example, a much better example when you actually take this information and you can see a very normal looking bell curve, right? And this is the reason for this better looking bell curve is because of some filtering. So let's take a look at how we can filter this data and what will happen to this histogram. Okay, so again, we have here our maximum value. So let us do the same for this. Well, actually, in this case, we don't need that, right? So we need to just, let's do a filter here. And let's set it to more than, greater than, let's say, this is in log scale, so let's do 7. Okay, and let's see what now happened to our data. Maybe 7 is too much. Greater than 3. Okay, let's leave it to think a little bit. Maybe I need to restart this. Okay, we'll actually do this in R later on. Might be a little bit easier to do quickly there. But the point that I'm trying to make is that once your information is more normally distributed, you can more easily understand the differences in your data. Now, another approach to not only do logarithmic scale, but also to address the comparison within each one of your group is to further normalize your data with what's called quantile normalization. Okay, what is the purpose of quantile normalization? Remember yesterday, we did some comparisons between genes in the same type of a sample, right? You remember that from yesterday? What we were looking for is a way to compare the overall values in our samples. Why are we doing that? When we did that yesterday, right, and I'll just quickly show you here an example from yesterday of what we did. What we did here, right, is the following. Let me just do this, right? We did a scatter plot. And what did the scatter plot show us? The scatter plot shows us a comparison between expression of a normal-like sample and a normal-like sample. So if both of these are normal-like, I expect my expression values to be highly correlated, right? We can further see that with actual correlation, right? So here we further then did something that looks like this. And we saw that, for example, normal-like and normal-like has a good correlation value. But if we look at normal-like versus basal, that correlation value is lower, right? So if we have our correlation value that is high, that means that these are indeed representative of this one normal-like uh, uh, group or condition. Because if I have replicates in my data, if I want to represent, let's say, smokers and non-smokers, and the variation between smokers and non-smokers is significant, I also have to look at variation within each group. Let's say if my non-smokers are all over the place, the data is very different for each individual, that will not actually tell me a lot about the difference between smokers and non-smokers. So when I prepare my conditions, when I want to compare gene data that represents a replicate that represents that condition, I want to make sure that they are fairly well correlated between them. And when quantile normalization, what it does is it actually takes all of the values in a particular sample and organizes them in the same order without changing the values, normalizing the distribution. Okay, so when we look at this, first we want to make this distribution a little bit more normal. Then we want to make sure that within our data set, the comparisons are going to be more according to same distribution, but based on the actual values that we have in our data. So when we do quantile normalization, as a result of that, we have, okay, right here you can see different distributions and different means. As a result of quantile normalization, you will actually get the same mean and the same distribution without changing the values, right? So it only changes the distribution and the statistical properties of our data. And also, we use a threshold because some of the information in our data is actually most informative. And which genes do you think are the most informative based on the previous assignment that we did? What are some ways that we can characterize informativeness of the genes? Based on the assignment from yesterday? different in different groups. That's one way. What's another way to just know that this gene is probably important? Exactly. So, well, not just in one type of cancer, but one type of cancer means you're already talking about a particular condition. But in general, genes with much higher expression are probably going to be the more significant ones, because if they are highly expressed, there's much greater chance that we will find a high difference. So if we find a gene, for example, let's talk about different tissues. What is the difference between, let's say, a liver tissue and a skin tissue? What is the difference? The difference is specialization, right? So these tissues all came from the same DNA, but the DNA code is being used in very different ways. In one way, it is becoming a liver, right? And it has a function of a liver, and that is determined by specific proteins that are being produced. And in another case, it's becoming a skin, and they're very different, even though they contain the exact same DNA. So we can measure those differences at the RNA expression level by looking at highly expressed genes. The highly expressed genes are the ones that are champions in that cell. They actually perform the main function. So when we have a gene that has a high expression, that is more informative, potentially, if we want to do any kind of downstream analysis, comparison, or anything else, that is going to be a source of greater information. On the flip side, the low expressed genes, why can we filter them out? Because the low expressed genes actually do not contain a lot of information. They probably play some supportive function that is common, that is very similar between any sample that we take. They're just responsible for maintaining some basic functionality that is common between all living things or all living cells in a particular organism. Now, in the middle, we have some bulk of our data, and that is really what's interesting to us, maybe, but there's where we need to find those significant genes. So when we do quantile normalization, we can take a threshold, and that threshold will actually help us eliminate the technical noise. So we want to eliminate the technical noise, that is the real low expressed values. In the middle, we will have what's called normal gene expression, the genes that are common between majority of whatever cells we take. And then the significant signal is going to be on this long tail of the distribution. And that is where we will typically find most of the information when we want to compare different signals in our data. So these two methods of log normal transformation, and which is really scaling of the data and produces a different scale. Instead of normal scale, it's going to be in logarithmic scale. And the second one is the quantile normalization, or there are other normalization methods as well, but in our case, we can rely on quantile normalization to really see that the power of a test, whatever power we assign to a test that we will perform downstream, is going to be more significant. So here's an example. Here are a couple of genes. You can see that it's the same gene actually before and after quantile normalization and log normal transformation. You can see that when we look at it in raw scale, we might be tempted to think that there's a difference between this condition and this condition. But the difference is primarily a result of variation within one group of samples, right? If we do quantile normalization and log normal transformation, you can see that that difference is practically zero. So if we find some difference, that is a result of this primarily, right? And this one sample is not representative of this condition. If we look here, that effect is eliminated. And so we are not going to be tempted anymore to consider this a differentially expressed gene. Also, it's important when we want to compare what we talked about, correlation within the same group of samples. So here we have one sample in raw scale. Here's another sample, or two samples from the same group, right? And you can see the distribution of your values is going to be much better when you look at it this way. Okay, so any questions about these aspects of data transformation and statistical summary before we start going into the code? Okay, if there are any questions, let me know. If not, let's go to code OmicsLogic. Yeah, one question. Go ahead. Okay, why do we do quantile normalization? So quantile normalization is a way to make samples within a given data set more comparable to each other. So sometimes you might want to say, I have two groups that are different. And the difference could be as a result of many different things. For example, one group of samples was prepared from larger samples or samples that have been somehow happier. They had more food, the environment condition was better, they were just more active. And so when I start to compare them, one of the things that emerges is that the difference is consistent. It doesn't matter which gene you take, any gene you take, that difference is consistent between two samples. If that is the case, and you don't want to go through all of your genes and test that case, but if that is the case, you're really finding difference as a result of non-biological difference, right? That does not represent the condition. That just represents the way that condition was measured. To eliminate those differences in measurement, you can make sure that the distribution and scale, both distribution and scale, are going to be the same. Yes. So what I asked about, homework assignment is to put it on the forum, and I just saw that Marek is the only one that put that in the forum. But you can all put it on the forum, and we'll actually review it here in a second. Right. To avoid bias and to improve the power of our parametric tests. In which form? So you can just explain what you have found, and if you have a screenshot, you can email that. Okay, so let's take a look at this assignment right here, descriptive statistics and visualization. So please let me know when you are in here, once you are in here. Again, we can do this in Python, but I really want to save that for a special session. We'll do that so that I'm not confusing everyone. So let's try and look at the R version. Okay, so please let me know when you are here. Okay, so 2 people are here. 1, 3. Let's wait until at least 5. Okay, 4, 5. All right, great. So we can skip this for now. The important thing that you can see here is that we load the data, then we want to clean the data, for example, exclude any kind of NAs or any kind of missing data, and you want to also filter your data to exclude zeros or maybe filter by a certain number, right? What we just tried doing in Excel and it got stuck. And then here you also have transformation to log scale and plotting a box plot, right? So here, further down, okay, so make sure that you have that library. Did you have this library installed, Daisy? And let me, okay. Okay, we can take a look at this. There's a number of ways that you can filter data, but we can look at that. I think it should work. Yeah, if you can put the report in the forum, that would be great. Okay, so let's go and look at the visualization after that. So again, we have an explanation of how to prepare a matrix, and then make a few different visualizations, a box plot, which we talked about, and a heat map. We also can do a histogram, right? So maybe it's not here, but also summary statistics. Okay, there's a histogram. Okay, so let's do this together. First of all, let's try it yourself right here in the code, just to make sure that it works. Okay, so you can open the console, run this. Okay, success running code. So we've imported this, and now we can try to run this and get some of these outputs. So what do we see in these outputs? Here we have our, what do you think is this data? Is this data how to complete last session's assignment 2? Yep, I will go through that. Okay, so I guess to reorganize then, if we want to do last session's assignment as matrix. So the matrix is, just your numbers. You don't have, and you have to enforce that all of your values are numbers. So that's basically what it does. Because a data frame can contain any kind of data. Okay, so here, what we did is we used this number. Now let's try, okay, if you can try, instead of this 15 genes, let's actually try to use the full data set and see what happens. Okay, so I'm going to give you a file. So let's use the cell lines expression FPKM. Okay, so you can use this file right here. So let's go to here and we'll just replace it with this file. Okay, what did it tell us? It tells us that it cannot allocate vector of size 1.5 gigabytes. And so that is a typical problem that you will run into when you start dealing with real data sets. To practice with real data sets, you really have to go on your own computer, and you have to have enough RAM on your computer to be able to work with these files. Even though the file itself is not that big, right? Let's see, how big is this file? It is 1.6 megabytes. But to work with this file, you actually have to have a significant amount of RAM. Okay, so that's why this isn't going to work. Okay, so because of that, let's try and work with this data set on your computer. Okay, and before we jump into this in our studio, I'll just give you a couple of minutes for everybody to open our studio. And by the way, in our studio, the reason why these assignments are going to be useful for you to complete first on a small data set is because you see how certain functions are being used. What are the parameters? You can change the colors, you can change the dimensions, you can change how you transform the data. But later on, if you start working with bigger data sets, you gotta have an environment where you can actually handle that amount of data. So how many of you have your RStudio open already? Okay, so Janani seems to be the only one. Yeah, how can you change your working directory? You go to Files right here, and you navigate to whatever place you want to be. Like, for example, this is all of my stuff, and I want to go to OmicsLogic Data Science, and then I want to go to my sessions, and then here I have session 3, I believe, is where I have all of my stuff. No, session 3, data. And now I have all of my files in here. I can go to more and set as working directory. Okay, and you can actually see what it does. Here it writes to you how it actually does it. Just a command set WD. And so if you know the path, you can just use that, set WD. Yeah, if you have a global working directory, you can do that. If your directory changes, you can change it from here. Okay, anyone else? Did anyone else get here? All right. So this is yesterday's, right? Remember, in that folder, you have, this was our first exercise, this was the second exercise, and this was genes introduction to genes, right? So what we wanted to do in this previous analysis that we did yesterday is we wanted to make sure that you understand how to extract information from the gene expression data, and how do you actually want to look at, let me just open this. How to deal with data that is non-intuitive, right? So what we first did is we loaded the data, right? So here we load the data, and here we can load the data instead of the 15 genes, right? For example, we can load cell lines expression data. So if I open this, I can then extract just the matrix, right? So just the numerical data from my data, and I want to visualize a particular gene, for example, gene 10, right? So I just take row 10 because that is all I have, because the top one is the header. And I want to visualize it as a bar plot. So to do that, I just run this. Oh, let me change it to 15 genes. Okay, so here, added all of that. Let's hide this for now. Okay, let's just do this. All right, so I get this bar plot. So did everyone get this for yesterday? I think we briefly went over it, but just to make sure, did everyone get this? Okay, for those of you that are working in RStudio, were you able to see this kind of a plot? Okay. Okay, so how do we know whether this plot is interesting or not? So one way we are trying to extract information about this gene is we want to see, okay, which samples is this actually being expressed in? And if you zoom here, you will see all of your bars mentioned. So you can see that normal-like and basal, right? Then you have luminal, they're pretty high. Then you have cloud and low is low, and then luminal again is high. So it's obvious here that if I change the order of my information, it will give me a little bit better sense of what's in my data. So to do that, okay, we can use this option to sort. And I can sort this by descending or ascending. And that will be based on the values, right? So based on the values that I have in this row, row 10, I will sort the data. So let's try and do this. So we don't need the bar plot, right? So let's just do this. And by the way, for those of you that are doing this, this is also something that would work on the OmicsLogic code, because we're still working with small data. Okay, so here now I can see that the information that this gene carries is the difference between luminal and the rest, right? The rest actually don't have a lot of variation because the basal is not together. If it was luminal and then basal were together and then the rest, then I would know that that will give me some more information. But in this case, it only carries information difference between all and luminal. Okay, did everyone get this to work? Just to make sure that everybody is still following along. Did anyone get this to work? Okay, so that's probably because of what you're using. So you can see what I did is I also made this as matrix, right? Okay, so let's go through this. So first we load, yeah, you can also do it on, okay, so here, let's do that in here. So let's do it in here. Okay, so here I can go to R, okay, and just copy the whole code. So just take all of this and copy it in there. By the way, where is this code, right? So if I just clear all of this, in this folder, right, you have this genes intro. So what you can do is you can just drag this over here and it will be copied in here. All right. So the first thing we need to do is we need to make sure that we tell it where is this data set. So this data set is not on the computer, right? This is not going to run off your computer, so you can't just put in the name. So where do you actually get the name or the link to this file? You can get it from right here, for example. Okay, so this is that same file. So let's try and replace it here. And let's run it. Okay. Oh, well, actually, we don't need this because we're not dealing with Excel anymore. What happened here? Yeah, we also don't need this. So actually, we just need this. Gene expression read table. One sec. Let's see. Let's just leave here. Yeah, genes intro R should work. Yeah. Let's delete all of this. HTTPS. All right, there we go. Okay, so if you just copy this, right, or it should be in there. Okay, so it should work, and you are getting, right, in this case, we first did this one, And here we just have the gene, or then we can also do the second one, right? So we get this gene, and then we get it sorted. Okay, so the objective of this exercise is to start asking questions. Okay, how can we find information from the data that we have? And so the way we can do it is by looking at specific rows. So let's look at specific other row. So let's take gene 1 instead of gene 10. And so here, obviously, if you take that, you have to change this. So change this. And let's do it sorted, right? Sorted is more interesting. So let's just compare the two. So we're going to just take this and we're going to do it one time for gene 1 and one time for gene 10. So this is gene 1 expression, and this is gene 10 expression. So which one do you think is more informative? Gene 1 or gene 10? Okay, anyone else? Right. Why gene 10? Why do you think gene 10 is more informative? Okay, again, this is not a histogram, this is a bar plot, and that's important to differentiate. A histogram shows you distribution, and the bar plot just shows you values, right? And so, right, so what we are really looking for is a pattern, right? Right, so if the pattern was half of them were 0 and half of them were like a lot, right? It is going to be more informative. But here what we see is more randomness, right? The pattern in this case is just defined by, this is more random, right? It doesn't actually provide us a clear differentiation between a particular type, which is described right here. So this is type, right? And this is the gene expression. And here, right, that pattern is very obvious, correct? So when it's really obvious, that means the information is more clear, right? So if I give you a noisy picture or a clear picture, how do you know that the clear picture is clear? It just, you immediately understand what's going on. And so in this case, this, you immediately, it comes alive. Okay, so did everyone understand this? Do you have any question about what the assignment was? Or does anyone have any questions about this? Okay, the problem is if we want to work here with the bigger data set, we can't, right? It's just too big for us to work here. So we have to rely on working here. So let's move on to the summary statistics, right? So how do we actually do summary statistics? So I propose here to do the following. We can have, and by the way, this is also saved in Dropbox, and that is called expression summary. Okay, what are we actually doing in expression summary? Let's take a new file, and we'll go through this together. Okay, the first thing that we want to do is we want to load in our data, right? How do we load in our data? So let's start like this. Okay, so let's take all of our genes and we'll say read table, right? And what is the data set that I want to work with from here? I want to work with cell lines expression FPKM. So here I type in cell lines expression fpkm that txt. Remember, I need to specify that there is a header. Okay, so how do I know that this file loaded? I type in view, all genes. So to run this, I do Command Alt R, and here is my data. Okay, is everyone with me? Or is anyone? Maybe if you have the data loaded, say yes. Excellent. Okay, so let's go back. What do we want to do next? So we don't need view all genes anymore, right? Because we know it's there. So now we want to just make sure that we remember the column names. So what are the column names? So let's just call it call names 1, and we'll take the IDs, right? So we want to take all genes, and we want to take ID, right? Because if we look here, this is called ID. So this column is called ID. So we want to take these gene names because we want to start visualizing these genes afterwards. Okay, so we'll just create this information here. Now we want to make sure that we take only the data part, right? Only the numeric values. So how do we do that? We can take all genes and only take, well, and by the way, this is an interchangeable thing. You can do equals or this sign. And we'll take all genes -1, right? So we don't want to take the top row. And we want to call it as matrix. As matrix, and then all genes, right? So we want to make sure that we take all genes. Okay. So let's see now, how do we, because we're going to be playing around with both of these, the log scale and non-log scale, I'm just going to create a data frame, right? And what I'll put in the data frame right now is all genes. How do I make log transformation, right? So let's take a look at all of our genes, right? And we can see here these numbers, they vary, right? You can see here 649, here you can see 8 or 2. So just by scrolling through 1092, right, you see that the distribution is very large between your small values and large values. So let's transform it to logarithmic scale. How do I do that? Let's create another variable called log data, and we're going to just make log of df. Okay, let's view our log data. Okay, so now we have our log data. So let's look at all genes. Here are all genes. Oh, let's look at our df. Oh, let me close all this. I'm not sure where this is going to go. Let's look at view all genes and view log data. Okay, so here's our all genes and here's our log data. How did it change? Okay, again, we have 914 here in the first one. Here we have 2 point something, right? Here we had, let's say row 21 is, or let's take row 41. Okay, you see the problem here is we're starting to get these infinites, right? Now, if you want to do this from here, there is, exactly, yeah, so we need to add +1. So how do you actually do that, you can see here a little guide. In here, I think. You see, log data plus 1. Okay, so remember that if you ever want to just see again where we're trying to get to, you will see it right here. So let's go in here, right? And so here we want to do plus 1. Let's try that again. Sorry, that's not the right one. That's in here. So here, df plus 1. Okay, so here's log data. Now we don't have any of those infinite numbers, right? What do we want to do with this data? Let's do a box plot, right? So let's do a summary statistic actually first before we start going into visualization. How do we do summary statistics? We just play summary. And we can do, let's do summary of log data. And we don't need to view these anymore. Okay, so here we have a summary for each one of our samples, right? You have the minimum, first quartile, median, mean, third, maximum, right? So all of this information, like for example, I can start seeing that all of my maximums are around 9, except for one of them. Right? So let's do the same, but let's do summary for, I guess, the raw data, all genes. Okay, what are my maximums? They're very different, right? Look at this, 15,000, 14,000, 8,000, 15,000, 13,000, right? 11,000, 7,000, 8,000. So my maximums are very different. And if my maximums are very different, right, if I just take some maximum gene, it can already have a lot of variation just based on something that I don't think is representative here of the biology. But this information is hard to read. It's just numbers, and I just see one number at a time. So how do I summarize this data visually? Again, well, let's do a box plot. Okay, how do I do a box plot? It's just, well, the simple version is box plot and of, let's do all genes. Okay, so there is my very simple box plot. If I want to do the same, but for log data, I can see the information is much better, right? So again, here I start visiting, and obviously I can make this plot a little bit better. You already know how to do this. For example, I can give it a name, right? So what is the name? What is the name? It's going to be like this. So the name would be, this is log transformed gene expression. Another thing that is bad about this is that I don't see all of my sample names, right? So here I can do class equals 2. Let's try that. All right, great. So now I have my name, and I flip these, and now I can see all of them. The problem is that they're being cut off, right? I mean, actually, if I go to zoom, yeah, it's still cut off. So how do I do that? We can set some properties. So let's do visualization properties so that I can just set some margins. So I can do margin equals, and here I have, 4 margins, right? Top, left, whatever. So I can do, let's do something like 14, 4, 2, 2. Okay, maybe I also, if I do it too much, this is going to be too small, so I can set also the font here. So maybe I can change here and make the font of the axis, so x-axis, right? Maybe let's do it 0.8. There we go. Okay, did everyone get this so far? Or did anyone get this? If you got this plot, say yes. Okay. Jerry John did. Cool. Okay, great. Anyone else? Does anyone have any questions about this? Okay. All right, great. Now let's do a histogram, right? So histogram is just hist. And let's do a histogram of log data. Okay, so there's my histogram of log data. Let's compare that to the histogram of all genes. Looks much better, right? So all data, or sorry, log data looks much better than all genes. Okay, so what can we do then with this? We can, of course, ask the question, just like we did in our previous genes, right? We can take a bar plot, so we can just copy this over from here. Right? And you can start asking questions about specific genes. So for example, if I have this bar plot that I want to create, I already have my data frame, right? So if I just keep this as data frame, and then I can just here, right? I can, instead of gene expression 1, this is going to be my DF, or actually this is going to be log data, sorry. So log data. And what colors? So we have some color here, that's fine. The name of this will be gene 10, but instead of gene 10, I actually want to get the actual gene name. So here, instead of this, what I need to do is I need to get my call names, right? Call names 1. Are you here? Are you looking right here? You have files and plots. If you open plots, maybe it's just not open. Try looking there. Okay, so call names and number 10, right? So what we want is number 10. Okay, let's try this. All right, so there we go. For gene ENSG, whatever, this is the plot. Okay, let's try to modify this, and let's do gene number 2. Anita, have you found your plots? Have you opened here, plots? There is a setting. Actually, the setting is like this. If you do a new file and you do R Markdown, right here, you will have somewhere one of these settings, preview images and equations, this is probably, and preview in window. So make sure that you have that. Maybe try that and let me know if that worked. Okay, so did anyone get this was able to generate a plot of your gene? Did anyone generate a plot of your gene? Anyone else? So here, you can copy and paste this, and I mean, later on, you can take a look again at the code. Did anyone else have their bar plot? All right, great. Did anyone else get it? All right. All right, obviously this is a useful thing, right? We can make sure that our data is loaded, we can transform it to log scale. Now we have a better distribution. We know that the information is good. Now, how do we actually visualize all of our data, right? That's the problem. Now, it doesn't really make sense for us to try and visualize, 20,000 genes, right? So instead, what we can actually do is we can take only a portion of our data, right? Let's say those 15 genes. So an easy way to do that is, instead of looking at each individual gene, we can just take cell lines, Cell lines 15 genes. And let's take a look at what that actually does. Cell lines 15 genes. Okay, and so here, obviously, we want to take, we don't want to do all of this. Well, we can do a box plot, we can do a histogram, we can do this. Well, we don't need this. Let's do a heat map. So just heat map. We want to use our DF, right? So all genes. Well, actually, okay, that's what we'll do is here, we'll do some, well, okay, we'll just do, sorry about this, all genes. Let's try that. Okay, what do we see here? This is like compressing a bunch of bar plots together and sorting them both ways. Okay, so actually a heat map does all of these things together. So here we have all of our samples. Okay, who got the heat map, first of all? Did anyone get the heat map? I mean, it's a very simple command. Okay, great. So you guys got the heat map. So what does this heat map actually represent? The heat map represents, like a bar plot, the level of expression. The level of expression is color. The darker the color is, the higher the expression. And it also organizes both our genes and our samples. So this is organization of samples, this is organization of our genes, organizes them in such a way that it is easier to read the pattern. Just like we organized our genes in our bar plot, it organizes it. And so what two main groups do we see here in terms of our samples? What two major groups are there? Okay, as you're thinking about this, I'm just going to change my my boundaries here. Yes. Luminal. Well, so what do these numbers actually represent? These are the genes. Remember, I have gene 1, 2, 3, etc. So these are my genes. And these are my samples, right? And so I'm asking, yeah, what two groups? So Maria and Mark correctly answered that I have luminal, right? Look, all of my luminal are here and the rest are here. So obviously, well, except for this one luminal, right? But here I have this major group, which is just luminal. And I have this major group, which is kind of the rest of them. What two or what three groups of genes do I have? I have a group from 5, 12, 8, and 7, this group right here, or no, maybe all of this, right? So this is 1, this is 2, this is 3. So all of these, and you can see the pattern visually is kind of different. Here the pattern is different, and here the pattern is different. So what we did in a histogram is kind of summarized all of this information together. Okay, any questions about this? Okay, does anyone have any questions? All right, if there are no questions, all of this information is summarized in Transcriptomics 2. So as you're waiting on your pipeline, right, you might not be able to complete your assignment in your Transcriptomics 1, start working on Transcriptomics 2. And on the code platform, you can go and obviously complete data visualization, but also you can do differential gene expression. Differential gene expression allows us to apply some statistics to find differences between two groups of samples. And you will see here, okay, let's see if this is, yep, loaded. What you will see here is that we are comparing samples from two types of breast cancer, ER positive and triple negative. And we are going to be looking at which genes, right? So from the total number of genes that we have, which genes are actually differentially expressed. And to find those differentially expressed genes, we can use a statistical analysis, a test, statistical test, and get a heat map of our results of the differentially expressed genes. So it might take a little bit of time, but you will see here the results, right? And here we have a result of the differentially expressed genes. And then what we'll do is we'll also create a heat map for this result. Okay. All right. Thank you everyone for joining today. Sorry, went a little bit over time, but hopefully. No, it should not ask money. BIPSA will look into it. It's just something that it wasn't added correctly. So we'll take a look at that, but it should be part of your membership. We'll go and look at it now. Okay. Any other questions? All right. Excellent. Thank you. I'll see you again tomorrow.